Hi, everybody. I'm here with the SEPM Moore Medal awardee, Dave Meyer, distinguished professor, lecturer, researcher, crinoid expert, and this year's Moore Medal winner. So, Dave, congratulations. And how are you doing today?
I'm doing all right. I've had kind of a rough year with various things, but I'm doing much better now, and
Good.
I'm looking forward to doing this.
Nice. Yeah, well, I'm excited to talk to you, ask you a bunch of questions, and for everybody else who maybe doesn't know you as well as I do, since we share similar research, but just to
Yeah,
get you to know you a little bit more, hear your
sure.
story, and what your interests are and what your
Yeah,
ideas are for the future of
yeah,
science.
yeah.
So, this is kind of a general question, but I think it's important for everybody to know how you got into your field, how you got into crinoids and studying
Right.
crinoids, and sort of how
Okay.
you got into geoscience.
Okay. Yeah. Well, when I grew up, when I was just a child, about uh, in the 50s, I got interested in fossils. And I grew up in upstate New York, which was loaded with fossils. So there were lots of great fossils nearby, the Hamilton group, the Devonian, the Silurian. And my parents really supported me very strongly right from the start. And I like to talk about my father taking me out on field trips, fossil hunting trips, on, say, like Sunday afternoon, instead of watching football like a lot of people do, he would take me on these field trips where we would collect fossils at outcrops that we learned about. And he once told me that if he'd been exposed to this when he was a kid, that probably would have been a direction he could have gone. As it was, he went into dairy science.
Yeah. Nice.
Yeah, that was
Did
his.
you? So that sounds awesome. And I mean, my father as well was super influential in kind of teaching me about the world and helping me grow and learn. And so going on these adventures with your dad, did you have a favorite formation or a favorite fossil that you like to find?
Oh, sure. Well, favorite fossil were trilobites
Mhm,
from the New York Devonian, the Hamilton group.
mhm.
That was the main hunting ground uh, at, uh, at where I grew up, and we had pretty good luck with trilobites. Not much in the way of crinoids. The
Yeah.
crinoids started with something called microcrinoids,
Okay.
and I got into this through my interest in having annual projects entered into the New York State Science Congress, it was called. And it was set up by geographic sections. We were the Central Western Zone,
Mhm.
which included Rochester. But but it was something where there was an annual 1 day science fair, and you could enter a display, or eventually you could enter a what they called a lecture demonstration, which was more like a professional talk in a way. You talked to the judges, came around, and you showed them your project, either fossils or specimens, and anything you wanted to show them and talk to them. They could ask you questions. Yeah,
So
that
you
that was a
focus,
good
yeah.
way to get started because you had to behave like a professional. Well, the way you would when you became a
Yeah.
professional.
Yeah. So was it at that point that you were hooked on crinoids and decided to
Yeah,
kind of devote your
yeah,
career to those organisms or?
yeah, pretty much. It
Mhm,
it went way back there because uh, one of my best projects was on so-called microcrinoids,
mhm.
and those were cued to me by somebody I met. His name was Bill Evatt, Evitt, William R. Evatt. He was a professor at the University of Rochester at the time. Very kind to us, supportive. We would visit him in his office, and he would talk to me for a while. And he gave me some localities, and he talked to me about uh, preparing material to examine for microfossils. And the microfossils I found were microcrinoids. And they came out of the Devonian Hamilton group. And pretty soon I had another colleague that I got connected with, a guy named John Koenig, Koenij, IJG. Jack Koenig was a geologist with the Missouri State Geological Survey, and he had done research on microcrinoids and published papers. He was extremely skilled, very, very painstaking man. He examined these things under binocular microscope, and he used the camera lucida to make drawings, hand drawings of these images on the camera lucida. And then he would do these drawings, and they would be stippled drawings. So they were really nice illustrations, and these got published in the Journal of Paleontology.
And you won a prize for that fair,
Yeah, yeah,
right?
yeah. I eventually, in my senior year in high school,
Mhm.
I won one of the top prizes in our science congress, and it ended up going to the state level, because if you
Yeah.
won at the section level, you
Uh-huh.
competed at the state level, which was down in Corning, New York. And I went down there and gave my lectem and won one of the top prizes there. So I ended up being one of the top 10 in New York State in that category for that year. And what happened was the top winners were invited to enter their projects in a special display at the New York State Fair, which was annual thing, and it was held in Syracuse, New York. This was no longer, you'd already won your prize, but you went there and you put on your exhibit and you met the governor. You met Nelson Rockefeller. So
Cool.
on Governor's Day, the governor comes around the fair and shakes hands with people, does the politician thing. And I got a picture of me with Nelson Rockefeller shaking hands. I got to like him a lot. I wasn't necessarily politically a fan of his, but I sort of became one. Rocky. And
Yeah.
and I felt he did an awful lot for education in the state of New York. He fostered the growth of SUNY, State University of New York, and that
Yeah.
was where the college was in my hometown, where I got my start from the contact with people there. That came
Yeah.
about because I went to elementary school
Mhm.
at Brockport State, which was a SUNY campus, originally a teacher's college.
Okay.
And they had a geologist on their faculty. His name was Victor Schmidt. And Vic Schmidt taught geology at the college at Brockport.
Mhm.
And he had a PhD from Cornell, and it had been done on not fossils, but he knew a lot about fossils, but it was done um, on glacial deposits. So he was basically a specialist in quaternary geology.
Yeah.
And uh, had studied at Cornell, got a degree, doctorate from Cornell. And
So,
it turned out down the road
mhm.
that Vic Schmidt was also a student, a Cornell product, and his let's see, he he had a course in which he needed a teaching assistant,
Mhm.
and Vic became a teaching assistant in that course. That was a course taught by Ken Castor, who later became a professor at Cincinnati. And he was kind of the guy responsible for my getting the job at
Yeah.
Cincinnati when that came along. So,
So,
so
so after, I mean, winning all of these awards and microcrinoids and meeting the governor,
yeah.
is an addictive feeling, and it's a feeling that makes you proud and even more interested in what you
Oh,
study.
sure, absolutely.
So then you
And
knew right away that's what you wanted to do going into undergrad and into college
yeah, that's true.
and like pursue a career in that.
Yeah. The microcrinates were a focus,
Mhm.
not because my dad and I had picked up a lot of good specimens in the field. Well, we
Mhm.
collected the dirt the broken down shale from the outcrops
Mhm.
and treated that and looked at it under a binocular microscope that my dad borrowed from his lab at work. And was very supportive that way. And so I got interested in microcrinoids and those were the first crinoids I ever collected. And they ended
Yeah.
up with this guy Koenig from Missouri.
Mhm.
He wrote up some papers on this stuff.
Mhm.
Some of the things we found were new species, and he described them
Cool.
and published them under with my name and his together. He wrote the paper,
Yeah.
but then he wrote another paper about the growth stages, which actually came directly from my project.
Nice. That's cool.
These things are only a millimeter or two high,
Yeah.
and they're just a calyx, but they're very resistant, and you can collect them by the hundreds. And so that was my project. And I could still do research on that. And I kind of have an idea to dust that off and
Yeah.
really do it right. Because I used it as a class project when I got to grad school and got a good grade for that too. So
Nice. So
yeah,
for those listening that maybe don't know what a crinoid is,
yeah,
it's an echinoderm related to
yeah,
starfish and sea urchins, and
yeah.
they've existed for I think is it 480 million years, Dave? Is
Yeah,
that
just about the lower Ordovician is when the first crinoids come into the
yeah.
fossil record.
And they
Yeah,
still exist today, which is really
yeah,
cool. And their
yeah.
morphology and anatomy hasn't really changed a whole bunch. And
Right.
so you studied modern crinoids all over the world, right? As a scuba diver, you dug down.
Basically, I got interested in in finding out what we could tell about these animals as living animals. That's the biology, the ecology, and so I determined that what I wanted to do graduate work on was crinoids as living organisms,
Mhm.
and to be because when you read the literature, where could I find these? You would almost draw a blank because there wasn't very much known at all. They had been discovered a long time ago. They were first collected by deep water dredgings, which nobody could do any diving then. That came after the Second World War.
Yeah,
And so we owed a lot to the development of technologies like scuba diving and underwater photography, I might add. That was
yeah,
extremely important too.
yeah,
So I got onto that, and the diving that I learned was was taken up for the purpose of doing research on crinoids. I was not first a diver, and then
yeah,
secondly, uh, interested in crinoids. I was interested in crinoids, interested in fossils and geology. And
yeah.
so eventually I wanted to go back to geology, to the
Mhm.
fossil record, and try to get more information that we could use for understanding the ecology of crinoids as living organisms. Yeah.
Yeah. Yeah, as a PhD student, you definitely need to adapt. And you learning how to scuba dive,
Yeah.
being from upstate New York, you definitely adapted a lot. And that was at Yale, is that correct?
Yeah, I did my undergrad work at the University of Michigan, had a great paleontology geology program, and
Mhm.
I worked with people there. Bob Kessling was an early mentor, and
Mhm.
then the second year I was an undergraduate, they hired Brad McCurda,
Mhm.
MACURDA, and I remember Professor Kessling saying, "Why don't you go see this new guy we hired?" To maybe he can help you. So I met McCurda, and I got signed up as a lab and field assistant with McCurda.
Mhm.
And in the summer of 1964, McCurda, with a grant, was going to do a European trip for research, and he asked me if I would like to go with him, and I was able to do that thanks to money that came from my grandfather, funded
Cool.
my trip to Europe with Brad. and
Yeah.
and we went for several weeks from the United Kingdom, Britain to the continent, and we did some field work. Mainly, we visited collections in museums. We
Okay.
spent a lot of time at the British Museum, about 5 weeks, And during that time, I was able to go through full open museum trays for all their fossil crinoids, which is one of the world's best crinoid
Yeah,
collections.
that's
And it,
fantastic.
yeah,
Yeah.
and meet a lot of people. The trip with McCurdy was meeting people and going places that were
Yeah.
well known and becoming acquainted with what we came to know as the echinoderm community
Mhm.
of researchers. And these included some of the very best people in the world
Yeah.
that I met at that time. And it also led to my working on living crinoids, you see.
Mhm.
That was because one of the gentlemen we met, a Dutch paleontologist named Bremer,
Yeah.
Breimer,
Mhm.
Bert Albert Bremer, he was a specialist on blastoids and crinoids,
Mhm.
and he'd published some very nice papers, a PhD on Spanish Devonian crinoids,
Mhm.
and we got to meet him in Holland. And I remember Herder regarded Bremer as a fine European gentleman, and that was certainly true. But he was a heck of a good scientist too. And
Yeah.
together they published a lot of very significant research.
Yeah.
Yeah. But it was Bremer who introduced me to one of the first studies I ever looked at that studied living crinoids in the field with scuba diving.
Mhm.
And that was a little paper published in a German publication, kind of like Natural History.
Mhm.
And it was published by the Senckenberg Museum. It was called Natur und Museum. You had to speak German, which I
Yeah.
did. I was learning German in college. And so I
Wow.
got to use that. And it was
Mhm.
cool because I ended up translating some of the papers.
Mhm.
And there was a German guy from Darmstadt,
Mhm.
a kind of a technische Hochschule teacher, which I gather is kind of a junior college type. I'm not sure just what it is, but
Mhm.
it was about studying feather star crinoids, unstalked crinoids in
Mhm.
the Red Sea with scuba, very shallow diving.
Uh-huh.
And it was published and it had underwater pictures. So that one paper taught me a lot. It taught me that I had to go learn scuba diving. I had to learn underwater photography and be good at it. And
Yeah.
fortunately, I had some friends at Yale who were excellent photographers who helped me by doing my photography and working with me in the field. Being diving buddies, that was another problem.
Yeah.
So one thing led to another, and I
Yeah.
ended up, picking a project on living crinoids for my doctorate. And
Yeah.
Yale liked it because we were developing projects in what we were calling paleobiology at the time.
Mhm.
And in 1975, the new journal Paleobiology was founded. Uh, and I had a very early paper in paleobiology. So,
Wow.
so one thing
Yeah.
kind of led to another. Yeah.
Well, it's amazing. Not only the places you've gone to do research, but just the people you've met,
Yeah.
and the opportunities you've had to meet
Yeah.
people and
Yeah.
explore new research, be one of the first papers published in this new journal,
Yeah.
is really fantastic.
Yeah.
Um, and so throughout your career, you've always gone back to the rocks, although you just said you studied modern
Right.
crinoids and in deeper
Yeah.
shallow
I
water.
had the interest in getting back to the rocks, getting back to the fossils.
Yeah.
And I had that interest, and I kept attending GSA meetings, giving talks
Mhm.
when I was a student.
Yeah.
And after I got a job, well, I continued to work on my living crinoids.
Mhm.
First in my PhD project, and then as a postdoc, I continued with a postdoctoral fellowship from the Smithsonian. So
Mhm.
I had a great affiliation with the Smithsonian, the paleontologists,
Mhm.
people especially like Porter Keir, who became director of the Museum of Natural History at the Smithsonian, and he worked on echinoderms. He was a specialist on echinoids, urchins.
Yeah.
But Porter, he was an interesting man, very interesting man. He became kind of a mentor to me.
Uh-huh.
He was one of the first actual people to be a diver to study living echinoderms, and he published a paper through the Smithsonian, which had one of the first pictures of a living crinoid taken by Tier underwater down in the Caribbean. So the
Wow.
Caribbean became my first study area for my PhD.
Wow.
Yeah, and
That's fantastic. Is that the Comanchuids, the
yeah,
non-stalk
these were feather
feather
stars. These
stars?
were
Yeah.
not stalk crinoids.
Okay.
At the time, the stalk crinoids were known only from very deep water, deeper than 100 meters,
Mhm.
and they still are pretty well restricted there.
Yeah.
But we've been able to look at them real close with the submersible, like the Nekton. No free diving to them as yet, but I'm sure some people can do it with the
Yeah.
technical capabilities
Yeah.
that exist now. So
They'll
I
learn
need
for their
another,
PhD.
yeah, I need another lifetime to do that in. But no, no, I never did any closed circuit scuba, any rebreather diving. I did all my diving with recreational scuba, compressed air,
Okay.
depths of less than 100 and 200 feet
Uh-huh.
of water. Yeah,
Yeah. Well,
yeah,
that's amazing.
yeah,
And so, you're still publishing papers. I know we've talked before. You have a couple
yeah,
new papers that are coming out. Out of all
yeah.
the research you've done, what's something, and it maybe not even research related, but teaching related, or just the friendships
Yes,
and
sure.
networking and people you've met? What are some of the things that maybe you're most proud of and that you're wanting to
Well,
share with
yeah.
everybody in your career?
Well, becoming a professional paleontologist who
Mhm.
was also trained as a biologist, because as an undergrad, I majored in geology. I got a degree in geology from Michigan, but I took some zoology classes. But then when I got to grad school, I went to Yale, and they encouraged us to take biology courses, real biology courses. And so I took ecology, I took advanced invertebrate zoology, and a very, very important thing I did early on was do a summer course at a marine station. And the marine station I went to was Friday Harbor Labs, a station belonging to the University of Washington in the San Juan Islands of Washington State. And I went there and took a wonderful class in invertebrate zoo, and they trawled feather stars from a site that they had found feather stars on along Vancouver Island,
Wow.
and so at first the first living crinoids I ever saw were the feather stars trawled up and survived, and we kept in aquaria at Friday Harbor, and I could study them.
Wow,
Eventually,
that's an incredible moment. Yeah,
yeah, yeah.
yeah,
Eventually, I got to scuba dive and actually study the living crinoids from Vancouver Island.
yeah.
But at first, those were the very first ones.
So it sounds
And
like taking advantage of all of these opportunities,
yeah, yeah.
going
You
abroad
you
to
had to be
Europe.
opportunistic.
Yeah,
You had,
absolutely.
networking was very important right from the very start. The people I was introduced to, I
Yeah,
was very fortunate. People like McCurda and Bramer, people who introduced me to other people.
yeah,
And very quickly, you find yourself working with some of the top people in the world. And
yeah.
I've been extremely fortunate that way. And I even met R.C. Moore when I was an undergraduate.
Wow,
I,
that's
yeah,
amazing.
yeah, I
Yeah,
met him.
yeah.
Yeah.
And then so talk maybe a little bit about your teaching career as well and your
Oh,
teaching
yeah,
path and
yeah,
yeah.
yeah. Well, my very first job after the postdoc was a job with the Smithsonian,
Mm,
uh, but it was at a station they had in Panama, in what was then still the Canal Zone.
mm,
They had some interest in starting a marine lab, and I was involved in that in 1970.
mm,
Yes, 70, 71. I did part of my postdoc going to, we call it STRI, the Smithsonian Tropical Research Institute. And it is today one of the very top tropical biology, both marine and terrestrial, in the world. I mean, the
wow.
STRI is very, very famous.
Yeah.
And so we had a lab that I worked at on the Caribbean side, right near the opening of the Panama Canal. And we did scuba diving almost every day there. There were loads of crinoids. That's what got me there, was
Yeah.
my friends told me that they'd been there and there were loads and loads of crinoids. And so
Yeah.
I went there. That became part of my doctorate
Mhm.
and was very rewarding.
Yeah.
So
That's fantastic. Um,
yeah,
and then some of your other colleagues, because you taught, well, when I met you, you had just finished teaching, I think at the University of Cincinnati, Ohio.
yeah, yeah, yeah.
Yeah.
That came along after I worked for the Smithsonian for 4 years.
Okay.
I've always said, well, it was supposed to be 4 months in Panama, turned into 4 years, because they
Yeah.
offered me an opportunity that I couldn't refuse
Yeah.
to work as a biologist
Mhm.
on a project that they were starting there called Environmental Sciences Program.
Yeah.
And the object of this study was to do long-term research on long-term change in the nearshore marine environment
Mhm.
of Panama. We could work on Atlantic and Pacific sides. We worked mainly on the Atlantic side and the Caribbean, very shallow water. And
Mhm.
we studied things like mass mortalities of creatures there due to low tide events, due to storms and things like that. And the project involved starting a lot of projects in environmental monitoring, which
Mhm.
seemed like very boring. A lot of people thought it was very boring, but
Yeah.
it was, we called it day-to-day data.
Yeah.
And the temperature of the water, how it fluctuated, how the solar radiation fluctuated, and
Yeah.
then we had biological monitoring, which eventually was fruitful because one of the things I studied was how fast coral was growing there seasonally. And
Yeah.
I studied the growth in the little branching coral that was very abundant there.
Mhm.
And we showed that they grew faster during the wet season of Panama than in the dry season. And that was a very interesting result, which actually never, well, we had to write annual reports for the Smithsonian, which went into bureaucratic oblivion. But I couldn't really publish real papers on that. But I was able to continue my work on living crinoids.
Yeah.
Yeah,
And so you carried that as then a professor at University of Cincinnati,
yeah,
like contributing your work and what you were doing
yeah.
along to graduate students that studied under you.
Yeah, that's right.
Uh-huh.
I got a job at Cincinnati in 1975. They
Mhm.
had a vacancy on the faculty, and
Mhm.
I was hired to be assistant professor of geology. So at last,
Yeah.
I'm a
You
geologist.
got
So
back.
what I'm really proud of is that
Yeah.
I think I've been able to pursue two tracks as a
Yeah.
professional, as a geologist, originally interested in fossils, and then returning to have the opportunity to do that, but
Mhm.
also as a biologist to study the living fossils, sort of like they are the crinoids,
Yeah.
and that's been one of my favorite goals that I think I've fairly well been able to do that. I'm very happy about that.
Yeah, that's fantastic. And
Yeah,
I've read a ton of your papers dealing with
yeah,
fossil crinoids and living crinoids.
yeah.
But some of the formations you studied all on the East Coast, correct? Or you also went sometimes out
Right.
west, right?
True, back to Friday Harbor, but
Okay.
most of the research. Oh, well, wait a minute. Yeah, the fossil work was initially in upstate New York and other
Uh,
land-based localities.
yeah,
But then the diving took place in the Caribbean, all over the Caribbean, and then
yeah,
eventually enabled me to go out to the Western Pacific, which is the real
yeah.
golden ground for living crinoids.
Okay.
Yeah,
And then your
yeah,
fossil crinoid work was like Tennessee, Kentucky,
yeah,
Illinois,
yeah, yeah, yeah,
right?
yeah,
Ohio, and then even
yeah,
New Mexico and in the Sacramento Mountains.
yeah, right.
Yeah,
Yeah, as an undergraduate, I had field experience and lab experience with fossil crinoids, working with Brad McCurt as an assistant, and
yeah.
eventually we became colleagues.
Uh-huh.
So mentor and student
Yeah.
became colleagues, professional colleagues, that it still goes on to the present time because Brad is 89 and still
Yeah.
going strong, still publishing too.
Yeah.
So
Wow,
he's
that's
been a
fantastic.
great role model, wonderful person.
Yeah, that's
Yeah.
amazing. Have
Yeah.
you, when you looked at, because you spent a lot of time looking at modern crinoids, both feather stars and stalked crinoids,
Yeah.
how was it then translating, looking at them alive today to then seeing
Yeah.
what remained in the
Yeah.
outcrop?
Yeah. What you could do from one to the other.
Yeah.
Well, at first, when I first saw living crinoids, feather stars, scuba diving, I was scuba diving. They weren't. They were alive.
Yeah.
But they at first didn't connect too well for me as to what we could do with them,
Yeah.
because they have no stalk. They only have an attachment stalk during the larval stage, and
Yeah.
then they're free living. Some swim. Most of them could crawl. But the there are also living stalk crinoids, which are very much like a lot of the fossils, and they are restricted to depths below about 150 meters depth in the sea. They go much deeper, but they're beyond scuba diving depths, at least for the present.
Yeah.
But they really opened our eyes, because in the early 1970s had a unique opportunity to go to Jamaica, where I'd done some of my shallow water work,
Mm.
and dive in the Necton submersible, a research submersible, little
Yeah.
yellow submarine. And this was brought to Jamaica by a geologist, Linton Land. People would have heard of Linton. He's a very excellent carbonate geologist and reef scientist. And he had a grant that enabled him to use the Necton sub and ship it to Jamaica. They brought it on the deck of a bauxite ore carrier, because right near the lab at Discovery Bay, Jamaica, there's a bauxite mine that it was Kaiser bauxite. They were the reason the lab existed in Jamaica at Discovery Bay, and uh, we went diving. We were able to dive in the Necton for a couple of days in 1972. And those were extremely important, because we went diving to hundreds, 600 to 1,000 feet, I'd say. And in that depth range, off the north coast of Jamaica, below the coral reef, you've got places where you get living stalk crinoids. And there's several species, and there you could, with the sub, you could get up close to them. You could look at them through a viewing port, very small, and you could take photographs with a handheld camera with a flash that was mounted outside the sub. So we got some good pictures. And on those dives, we were able to see the way these crinoids lived. They the way they function was to take advantage of natural currents that swept their living environment, and they would filter water from that with what we came to call a parabolic filtration fan. The
Yeah.
filtration fan is the way crinoids gather their food. They don't just capture stuff dripping down from the surface waters. They don't feed that way. They feed from horizontal currents. And so I became real interested in how crinoids and other similar invertebrates utilize the water natural flow,
Wow.
and included that in my PhD and in my
Yeah.
later research.
Wow. That's
So I'm
fantastic.
big on currents. As long
Yeah.
as they're not too strong, as long as they don't sweep me off to somewhere I don't want to go.
That's
Yeah,
right.
yeah,
Yeah.
yeah,
Well, that's fantastic. Like learning how these crinoids live today and maybe translating that to the rock record and
yeah,
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 that's fantastic. Um, so a couple questions now, maybe not shifting gears, but what advice would you give to, I don't know, I mean, you've done it all from by the time you were a kid until,
yeah,
really into your career. But what advice would you give to maybe a student in undergrad?
Sure.
Yeah, not knowing.
I'd say go for it,
Yeah,
because I think we've barely scratched the surface or barely
yeah,
dived below the surface.
yeah,
There's so much. And
uh-huh,
in my research, I developed the use of photography, underwater photography. I had some excellent people helping me. Bill Sacco, a wonderful professional photographer with underwater photography, worked at Yale. And we were able to start to use time lapse underwater photography, where we used a camera that would take pictures in a time lapse sequence day and night,
mhm,
illuminated by artificial light from a flash, and take very good pictures, which we were able to publish. So I became a real fan of direct observation of these animals and
yeah,
study of them by by time lapse and still photography, and also movies too.
yeah.
And,
So
and
just take all these opportunities to
yeah,
learn new skills,
yeah, yeah,
meet I guess the right people or just
yeah,
a bunch of people, right,
yeah,
throughout your career.
yeah,
To take those opportunities, say yes to opportunities.
yeah.
Um, and then just kind of more recently as we kind of end the interview, I was just thinking what's your take on how the future of geoscience is sort of progressing, uh, and how we can kind of maybe,
Well,
enhance
yeah,
that?
sure. Well, I consider myself extremely fortunate to have grown up in the science of geology at this time from the 60s on, because during that time, when I was a grad student, plate tectonics was becoming into the mainstream. It was no longer an oddball thing that people were skeptical about. It became widely accepted and adopted as plate tectonic theory.
Mhm.
And that, being at Yale. You had people coming through giving talks. Some of the top people in the field gave seminars for us that made a big impression on me. So
Yeah,
so uh, so that was an important thing. And then I mentioned paleobiology was kind of a
yeah,
craze
mhm,
spreading through paleontology. People were getting interested in more of the kind of thing I was doing. There's other people who have pursued a similar faith. You've got people like Peter Ward. Peter Ward is well known as a guy, a paleontologist who studied the living nautilus, the swimming
mhm,
cephalopod. And then I later met Bruce Saunders, who did wonderful research by tagging and marking and recovering tagged individuals in the Pacific and Palau.
yeah,
And I was able to scuba dive with him, help him a little bit when he was working with Nautilus.
yeah,
Uh, Pete Ward has became a very famous scientist working on the biology of Nautilus. But again, these guys were initially paleontologists and they went back to it.
yeah,
So I'm not the only one, but I was one of the early people doing this.
yeah. And
Uh,
so you've seen a lot of change throughout your career,
oh yeah,
right? And this
yeah,
vast progression of these
right,
sciences.
right.
Yeah. What do
I
you think
mean,
would be a way we can, take that progression and continue that progression into the future?
well, by trying to pick up on the very latest developments in exploration,
Mhm,
which include time-lapse underwater photography,
mhm.
include video, of course,
Yeah,
uh, remote cameras, all of the things you see on TV documentaries about marine biology.
yeah,
Um, yeah, we a couple of years ago we put on a workshop with the Paleontological Society about underwater studies of crinoids and the use of time lapse video and that went over pretty well, I thought. So
yeah. Well, there's an amazing video. I always go back to it. The crinoid crawling on
yeah,
the
yeah.
bottom of
Well,
the seafloor in the
some of my
Bahamas.
colleagues
Yeah,
actually observed that stalk crinoids are not fixed attached by their stalk their whole life. Some of
yeah,
them break off they can crawl around. They are not actually permanently attached either,
Yeah.
because they can crawl around with their arms, and they leave trails that
Yep.
people think they can see as trace fossils.
Yeah.
And it's amazing. They've also found that certain types of sea urchins are predators on these
Uh,
stalk crinoids. They go after a stalk crinoid, they'll
yeah.
take them down, they'll start to punch on them, and they prey on their cousins and the echinoderms that way.
Yeah.
So it's amazing the things we've learned about the biology of crinoids through
Yeah.
these approaches.
Yeah.
Yeah,
That's fantastic.
yeah,
Well, Dave, do you have any other closing remarks to maybe students watching this, colleagues out there,
yeah.
old PhD students you've had,
Oh
or
yeah,
anything that you'd like to kind
yeah.
of close
Well,
off with?
it was kind of difficult to get PhD students to work on living crinoids because you had to be a scuba diver. Really, you had to be some kind of a diver. You had to have the dedication, the interest in these animals as a career. And but I was very fortunate. I had some excellent students at Cincinnati who were able to do this and do some wonderful research and
Yeah.
become professionals.
Yeah.
Uh,
Is
so
there anybody that you want to thank particularly for some of your achievements? I know you mentioned your parents early on.
yeah,
Uh,
Brad McCurda,
yeah.
especially
Uh,
people I've collaborated with, Charles Messing,
yeah.
Messing, a biologist who studied crinoids for his doctorate at University of Miami in Florida. And unfortunately, he had brain cancer, and we lost him a couple of years ago. But he did some wonderful research. One of the things he did that I think is an all-time record study was a time lapse of stalk crinoids in
Yeah.
a population located off the Bahamas in about uh, 400 meters depth uh, uh, off Grand Bahama Island. And
Yeah.
there's a whole community of crinoids there that has persisted there for years. And through an NSF funded study, he was able to use some very sophisticated underwater photographic material equipment that would take time lapse pictures at that depth. And the site could be relocated with locating devices. And
Wow.
so they made a time lapse study over a 3 month period of this population and made a time lapse video. To me, it does everything because it's the behavior of the crinoids with changing current. It's the population, the community ecology. It's everything you'd ever want in a
Yeah.
study of a living animal that was also important as a fossil. So
Yeah.
if you want to see what crinoids are really like, you go look at that
Yeah.
video online. Yeah.
Yeah. Well, that's fantastic. Dave, hearing your story from, starting as a kid, just digging
Yeah.
around in the rocks, taking
Yeah.
all of these opportunities, really meeting all these fantastic scientists, and
Yeah.
while also advancing your own career, to having
Sure.
students along the way that really admire you. Congratulations again on the Moore Medal.
Well,
I know
thank you very
everybody,
much.
yeah, thank you. And
Yeah. Thank you
everybody is
very
so
much
proud of
for,
you and the work that you've done.
I really appreciate the opportunity to talk about the work I've done. I'm still trying to do it,
Yep.
shop till you drop type of thing. Uh, if I was given another lifetime, I'd gladly do it all over again. And yet this time I might learn to scuba dive and tickle some of them stalk crinoids in deep water.
That's
I know there are divers who can do that.
yeah,
I'm not trained for that, but I'm sure there are people who can do that if they would. And
yeah.
I can tell them what to do, and I can stay on shore and enjoy the divers.
Yeah.
Yeah.
You'll be on the beach with a drink in hand and a water watch.
That is basically it. Yeah. Yeah.
That sounds great. Well, congratulations again, Dave. We're really so proud of you, and thank you so much for talking today.
Well, thank you very much for this opportunity and
Absolutely.
to the SEPM for the award. And I mean, that's how these things can inspire people, I think, and I hope they do. Yeah.
Yeah, I agree. Absolutely. Yeah.
Okay.
Thank you so much,
And
Dave.
meeting you, Matt.
Yeah, I know.
Meeting people like Matt and his colleagues in the field he's doing are right out there on the forefront, and they're going to be the future of fossil crinoid studies and how
Yeah.
they build the rock record for us. Yeah,
And making great connections and being open to those
sure,
connections. Absolutely.
sure.
And
Yeah,
passion. Yeah, being passionate about those things.
yeah, yeah,
Yeah.
yeah.
Yeah. Once you go
Thank
down the
you very
crinoid
much.
rabbit hole.
That's right.
Yeah. Well, take care, Dave. Thank you so much.
Okay, thank you guys for doing this for us, and
All
we'll keep up with it as long as we can.
right.
Yeah.
Thanks.